Okay. So again, please, if you have any concerns about your exam or the performance up until now, I'm happy to talk to you and look at things and see how things are going for you. There's a lot of points left out there for grabs. We have a quiz this Friday, and then you still have all your lab reports and then the final. All right, so there are a lot of opportunities left out there. If you did well in this exam, good for you. If you didn't do as well as you wanted, it's not the end of the world. There are other opportunities. Okay, let's pick up where we left off. So, how do we measure total carbohydrates? Haley. Exactly. This is the method that you're doing in the lab this week. It is not capable of determining all complex total carbohydrates you have in your sample, and it's usually used if you have research purified samples or beverages with limited types of carbohydrate in it. It's not a good method for a system that has starch, dietary fiber, oligosaccharides. It's not really the method of choice. Okay. What are the preparatory steps for carbohydrate analysis? Go ahead, Aya. Fine. Yes. So if the sample is not a dry powder or a flour already, we need to remove the moisture. And definitely if you have fat over, sometimes even if you have low fat content, the first step is defat to remove interferences. Very good. So we talked about extracting monodi and oligosaccharides. Does anybody remember? For measurements, if you have a complex sample, you want to get the monodi and oligosaccharides out so you can characterize them. How would we do that? Do you remember? We solubilize them in 80% ethanol and with a little bit of heat to enhance solubilization. And we get them out, we remove the solvent, and then we reconstitute them in the solvent for analysis. What are some of the methods used to determine monodi and oligosaccharide? I guess we covered just one type of methods, the reducing sugar method. We covered principle. The smoothie Nelson is the most common one. And all three methods we talked about are based on reducing cupric to cuprous by reducing sugar. And the reagents are different among the methods, and also the method of detection might be different. So some methods of detection is by weighing gravimetrically, other by spectroscopy, other by titration. Okay. So we stopped. We didn't go over here. We just did the reducing sugar methods, and then other very common types of analyses we do for monodi and oligosaccharides are enzymatic methods. So you already had the use of enzyme in food analysis lecture. Make sure you study it well. It's very relevant to many types of analyses. And here's an example of the use of enzymatic methods. So it's very popular method, like I said, specifically for carbohydrate. Enzymes are biological catalysts and they have high specificity. So we can use enzymes to specifically measure glucose or specifically measure maltose. Well, usually maltose is you hydrolyze it to glucose and you measure glucose. So they're very specific to the carbohydrate of interest that you are trying to analyze. So they're very effective. So usually when we have enzymatic reaction, like you heard Suzanne Nielsen in the guest lecture, you're measuring either the substrates or the products. Oftentimes you measure the product because your reaction leads to a product that absorbs light. And you can use spectroscopy oftentimes to measure the absorption and use standards and convert that to quantity. So, and very common enzymatic reactions are the coupled enzymatic reaction that you guys heard Suzanne talk about. So the first reaction is auxiliary reaction, but it's also called measuring reaction, where your food constituents of interest reacts with another reagent that you put in, and you get products. Neither of these, all of them, the reactants and the products, don't absorb light. That's why a coupled reaction is needed. When all of your reactants and products do not absorb light, we need something else. So we take a product from the measuring reaction and we couple it with another reagent to give us a product that has an absorption and can be measured. So that second reaction, we call it an indication reaction or indicator reaction, because that's where we generate a product that can absorb light and can be used for measurement. This is very common. A lot of analyses involving enzymes are coupled reactions. So an example is measuring glucose. There are two different types of reactions to measure glucose. This is the less common one. We'll talk about another common one later. But here, the glucose is reacted with ATP. The first enzyme that you use in the measuring reaction or auxiliary reaction is hexokinase. So the hexokinase removes the phosphate group from ATP and link it to glucose, so you get glucose 6-phosphate. One of the products here is the glucose 6-phosphate that reacts with NADP plus in the presence of another enzyme called glucose 6 dehydrogenase. So meaning we're removing a hydrogen from glucose 6-phosphate and giving it to NADP to get NADPH. Now, NADPH absorbs light at a region where NAD does not, or NADP does not. So we measure the absorption of this NADPH and trace it back to glucose using standards. So that's one example. There are other applications, like I said, glucose, galactose, even lactose. Well, there is maltose, sucrose as well. And then we use enzymatic methods to determine polysaccharides, total starch, amylose, and then we can get amylose to amylopectin ratio. And we'll talk about how we do it later on when we cover starch. So there are many advantages to using enzymatic methods. First of all, there are a lot of kits available for us. So we just purchase the kit and it comes with instruction and we follow instructions. And even they tell us how to do calculations and it's very easy to follow and usually very reproducible. And it has high specificity to the substance or the constituents we want to measure. And low detection limits, that means low threshold. So we can detect at very low concentration. Now, everything has disadvantages. So basically, you might have components in your complex matrix system that absorb at the same wavelength. So we try to correct with using correct blends. And sometimes you have to do the standard addition process, like we talked about in one of the spectroscopy lectures. Another issue with the enzymes, they might not be 100% pure. So you might have side reactions happening due to impurity of the enzyme. That's what it means by not 100% specific. But in general, they are robust and can be done in short periods of time and reliable. Okay. So we are still talking about mono and oligosaccharides, right? So far we talked about reducing methods. So colorimetric reducing method. Second, we talked about enzymatic. And now we're going to talk about chromatographic methods. So paper and thin layer chromatography are just honorary mention. We're going to mention them very quickly and remind you that these are qualitative. You can do semi-quantification by looking at the intensity of the different spots, or also you can scrap off the spot from the thin layer chromatography plate and use another quantification technique. But mostly it is qualitative. And these are FYI, you don't have to remember much here. It's just giving you an example of what would be the stationary phase and the mobile phase in paper and thin layer. I'm going to spend a little bit more time talking about HPLC applications. So we spent, I don't know, 5, 6 lectures talking about chromatography. And I told you, I promised you that we'll come back to it when we look at different characterization and analyses. And here we are, carbohydrate and mono and oligosaccharides. We use chromatography or HPLC and also GC, I'll talk about it later, to analyze, detect, and quantitate mono and oligosaccharides. We use normal phase, reverse phase, anion, and cation. These are the most common HPLC. I would say they are common, but if the most really common is normal phase. Most of all of them is normal phase. But the others are also used, and it depends on what you have in the lab and your expertise that you have. So I'm going to talk about each one of those, how they apply to mono, di, and oligosaccharides. All right, so normal phase. With normal phase, if you use unmodified silica, it's really not very good. If you just take unmodified silica, it's normal phase, but it does not give you good results, doesn't give you good separation because of the very strong interactions of hydroxyl groups with the silanol groups. So not used commonly. What is used is a modified silica. And the modified silica that normally goes with monodiene oligosaccharide is the silica that is modified with amine group. It will show on the next slide. But what I want to remind you is that modified silica makes the normal phase helic. You have to go back and review helic so that you see the connection here, which is hydrophilic interaction liquid chromatography. It's a form of normal phase where the silica is modified. Modified in this case with amine group. So the amine group here, so this is your silica, and you have kind of an arm, a hydrocarbon arm, and an amine group. So your carbohydrates, monodi and oligosaccharides, are forming hydrogen bonds. So the OH groups of your sugars are forming hydrogen bonds with the amine. Okay, so this is how they interact, and they separate based on the number of OH groups you have in your sugar. So if you have glucose, sucrose, and give me another oligosaccharides name, raffinose. If you have glucose, sucrose, and raffinose, which one will you use first? Don't look ahead, just look at me and think about what you see here. The interactions with hydroxyl groups. with the column. So if you remember your chromatography, try to tell me which comes out first, the glucose, the sucrose, or the raffinose. Will come out first. Hold on. Raffinose will come out first. Well, wait, go ahead.
I was gonna say, if it isn't that, that's gonna be sucrose.
So what do we have in the mix here? Glucose, sucrose, raffinose, mono, di, and tri. So it's either the tri or the mono. Which one do you think will come out first? Let's take the raffinose. We could change your answer to glucose. That's all. Correct. Because glucose has less OH groups than your oligosaccharide. So the more OH groups you have, the more binding sites you're going to have with the column. Because you have OH interacting with amine. The stronger the interaction, the more delayed they are. Remember that part of chromatography? You need to have interaction with the column to get separation. And the least interaction leads to elution first, and the most interaction leads to elution last. Okay. So that's the order here. Fructose, glucose, saccharose, lactose. So why fructose come out before glucose? It's a pentose. Fructose is a pentose, glucose is a hexose. So its structure limits its interaction with the amine groups. All right. So usually we have refractive index detection. When we have refractive index detection, if you remember, we mentioned it in chromatography, we cannot have gradient elution. Why? It has to be isocratic, same concentration of the mobile phase. Usually it's water or a buffer. Why? Yes. Exactly. So if your components of the mobile phase is changing, it's going to impact your refractive index. So you will have convoluted results. So you need no interaction or no impact of the mobile phase. You might want to have it constant. And then any change in refractive index will be due to your constituents. So the only drawback is if you have reducing sugars and you have amine, over time you might have this Maillard reaction happening and blocking of amine groups on the column. You can fix it easy by adding modifiers to regenerate the column. So basically you can add amine compounds with amine groups to regenerate your column that you will then be able to use that same stationary phase again. There are other these are just other examples, FYI, of modified silica. Remember the amine as an example, amine modification. Reverse phase. So you can use reverse phase HPLC, but it's not really very common unless you don't have another choice. The reason it's not common, remind me again, in reverse phase, what's the stationary phase, polar or non-polar? Non-polar. So is glucose polar or non-polar? Polar. It will not like to interact with your C18 column. It will just want to come out very quickly. And then if you have a lot of monomers, you will not have good separation. They will not separate, they will not interact very well with the column. So it's really not great. Unless, if you don't have any option, you can add a little bit of salt in your mobile phase. Then you shift the interaction of your monomers. Instead of the mobile phase that has salt, they will be retained a little bit more on the column. Now, another issue that I have here is that you have isomers sometimes anomers, we call them, of some monosaccharides. If you add an amine group or an amine compound, it will shift them to one form. Then you won't have split in peaks. But that's TMI. Yeah, Gen Z's, what's TMI? Too much information. Okay. So you don't have to remember that one, but I want you to remember that reverse phase columns or reverse phase HPLC is not very common for monodienoligosaccharides unless you add a little bit of salt so that you enhance the interaction with the column. Okay. Anion exchange chromatography. So remind me again with anion, what does it mean? What is the charge on their stationary phase? Positive. Thank you for remembering that. So we are exchanging negatively charged molecules. Now, is glucose charged? No. So neither is sucrose or lactose or any of the carbohydrates. They are very, 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 very weak acids. So in order to have them carry a negative charge, I would have to use a mobile phase with very high alkalinity. So very high pH, then you will force them to lose a hydrogen or a proton. So that's the only way you can use anion exchange is at really high pH to force them to lose their hydrogen proton from OH groups. And because it's charged, you can use these two detectors we talked about in HPLC, pulse amperometric detector or electrochemical detector. So order of elution. So glucose, fructose, lactose, sucrose, depending on their structure and the interaction with the negative charges that you have. So the more H is lost, the bigger the sugar molecule and more ability to lose a proton at a particular pH, the more interaction you have with the cup. So this would be the order of elution. Lastly is cation exchange. So there's no way you can have them be positively charged. Even at very, very low pH, they will not gain a hydrogen. They cannot be on their own positively charged. So this is the last you would go to unless you have capability in your labs to do it. And that's the only method available to you. And the only way to have them carry a positive charge is to introduce a cation. So either divalent metal ions or dilute acid that you have protons, positively charged protons. So your carbohydrate will just interact with the positive ion, and then they will carry the positive charge and separate. Usually this kind of chromatography have a combination of ion exchange and size exclusion. You can separate based on size and charge for this particular application. Now this is least common for monodi and oligosaccharide analysis. So yes, Claire.
There like an actual application an ion
for that?
Yeah, but
so you can analyze any of monodi and oligosaccharides if you have the system in your lab. Again, if you remember, many methods can work for the same thing.
like nobody would actually use it because
Unless your lab is set up with a system that they already have this things that can separate based on size and charge, then you can use it for your problem. So a lot of people are trained on different methods, and it's just a matter of what's available and who's trained on what. All right. So GC, are carbohydrates volatiles? No. So what do we need to do? What did you do to your fatty acids? You derivatize. Yes. You derivatize them. So you derivatize the sugars, and usually you have multiple ways of derivatizing them. Silylation or acetylation, or even trifluoroacetylation. Different ways. Again, Graham, what's available to them, what they're trained on, they will utilize. So first, they reduce the sugar to sugar alcohol. So they break the sugar into sugar alcohol, and then they apply the derivatization. Now, in this case, acetylation. So now we have a compound that can be volatile and thermally stable. And then we can apply GC. Very common used detector is FID, and this is FYI, the type of column used. And you can use internal standards. Here in this case is inositol is your internal standard. Okay, any questions? All right. Polysaccharides analysis. So we're going to talk about starch and dietary fiber. Under starch, we're going to talk about total starch determination. And then these two characterizations are very important in the food formulation world and shelf life and development of products, looking at degree of gelatinization and degree of retrogradation. So for starch, you can do qualitative analysis, such as just knowing if you have starch in your system, you can add iodine, turns blue. So oftentimes in analysis, we'll see it next week in the lab for fat characterization. We use starch as an indicator when we have iodine in the reaction. It will make sense next week. But in terms of starch, knowing the presence of starch, you can add iodine and then you will see if it turns blue, purplish blue. You can also use microscopy to look at the morphology of starch or the different starch bodies and structure. You see that every type of grain, for example, have different starch bodies. The way to determine total starch is enzymatic. So first of all, when you have starch, you want to measure total starch. The first thing you want to do is chop it off all the way to glucose. What are the enzymes without looking into the next page? I don't know if I have it in the next page or not. Okay, Gray is looking into the next page. I said, without looking into the next page, and
Ray's ready to answer the question.
what are the enzymes that Gray start? Did you look or not yet?
No, I did not.
Oh, yeah. Alpha amylase, that's one. What else?
Beta amylase.
Beta amylase. Which one is exo? Which one is endo? Huh?
Beta is exo.
Beta is exo, look at that. And alpha amylase is endo. And beta amylase breaks 2 glucose units at a time from the outside, from the external of the chain. What else? Glucoamylase. What does glucoamylase do? It breaks 1 glucose unit at a time. Okay, so this is very important enzyme because alpha amylase is gonna chop internally, breaks into multidextrin, different sizes. 3, 4, 5 degree of polymerization. But the glucoamylase will break one glucose at a time. So once the alpha amylase breaks the whole into smaller chains, then glucoamylase is very important to break everything into glucose monomers. So here's that. This is a figure that was developed by a colleague of mine. We started working in the University of Minnesota 15 days apart. After 5 years, he went back to Germany, but his slide lives on. Yeah, so I didn't steal it, he gave it to me. So, but he was the carbohydrate chemist. So, anyway, so this is kind of a structure of an amylose or an amylopectin. So you have the alpha-1,4 linkages, which you probably studied in food chem. The glucose are linked to each other via alpha-1,4 linkages. One, that means carbon number 1 and carbon number 4 from each of the glucose. And then you have the branches that is alpha-1,6 linkages. So that's carbon number 6 of one glucose and carbon number 1 of the other glucose. So you form those branches. So the different enzymes are specific to these kind of glycosidic bonds. So you have, first of all, the alpha amylase can cut alpha-1,4, and it sometimes can cut also alpha-1,6. So, but polymerase, for example, is a very specific enzyme to alpha-1,6, to the debranching. This we call another name for it is debranching enzyme. And then you have the beta amylase that cuts 2, sometimes 3 units from exo, so from the side chain. And then the glucoamylase, 1 glucose at a time. So when I want to measure total starch, I want to make sure I have alpha amylases there that cuts. Sometimes it can cut also alpha-1,6. Sometimes you add polymerase, sometimes you don't. And then I want to make sure I have glucoamylase because I want to break everything into single glucose units. Now, one thing I want you to remember, which will come back later, is beta amylase and polymerase only work on gelatinized starch. Cannot hydrolyze raw starch. So, or retrograded starch cannot act on resistant starch. Beta amylase and polymerase. Okay. Then once you have the glucose, here's another example of enzyme. This is the most common one that comes with kits that you buy. So you break down your starch into glucose, and then you use coupled enzymatic reaction to determine glucose. So in this example, you use glucose oxidase. In the presence of oxygen, you produce a product called gluconolactone. We don't care about it. What we care about it is in the process, we produce a peroxide. This is what we care about, and we add a dye. This peroxide in the presence of peroxidase will oxidize the dye and give us a colored dye that we measure using spectroscopy. We run a standard curve with pure glucose, and we construct a curve, and then we convert to total starch. Any questions? Okay. So now the degree of gelatinization, degree of retrogradation. So remember the very first thing I asked, can you use the same method to determine both? Or one or the other? The answer to that is yes. You use the same method. So, but you always have to have your reference or control. either your raw starch, and after applying certain processes, you want to determine how much of that raw starch got gelatinized. Or if you have gelatinized starch and over time you want to determine how much of that gelatinized starch got retrogradated. So you always have to have your reference sample, either raw starch or a gelatinized starch. Okay, so in this assay, you specifically use those two enzymes that I told you will only act on gelatinized starch. They cannot act on retrogradated starch. So what happens is you get your reference sample and you have your sample that you tested. You add to both your polonase and beta amylase. So the polonase is going to debranch, so you get linear segments. And the beta amylase is going to break, is an exoenzyme, will break dimers and trimers. And then you will have those maltose and maltotriose, they're reducing sugar. Then you can use a reducing sugar assay, like Smoothie Nelson, and determine the reducing sugar that you have generated. Now, if you have high value, does it mean it starch is retrogradated or gelatinized? Please think about it. Just to give me hope that you were listening. If I have more of reducing sugars, does that mean the starch is retrogradated or gelatinized? Because these two enzymes only act on gelatinized starch. They won't function with retrogradated. So the more retrogradated starch you have in your sample, the less reducing sugar you're going to get. The more gelatinized starch you have in your sample, the more you're going to generate maltose and maltotriose. So opposite results of your reducing assay for retrogradated versus gelatinized. I can repeat if you want me. Okay, yes. So the key point that you need to remember then, these two enzymes, they do not act on retrogradated starch. If you remember that, everything makes sense. So these two enzymes don't work on retrogradated starch. So you bring your starch sample, you have a reference. You always need a reference. You add those enzymes. One is debranching, one will cut to maltose and maltotriose. If you have, and then you do a reducing sugar assay, smoothing assay. If you have high value, that means the starch is mostly gelatinous. If you have low value, that means the starch is mostly retrogradated or resistant starch or raw starch. Huh? The way to determine, you really have to have your reference starch and compare to a reference sample. Is that good, Elisa? Did we capture that? What, did you lose me somewhere? So if you're building viscosity and you want to determine what's the heat time combination that you need to use for cornstarch or wheat starch to get to a specific gelatinization. So you do an experiment, the heat time combination. After that, you take that sample and determine the degree of gelatinization. And you compare it to your starting material before you run your experiment at, let's say, 90 degrees, 80 degrees, 10 minutes, 5 minutes. Kind of like figuring out what processing parameter you need to do to get to a specific viscosity when you change your starch. Sorry. Did I do a good job? All right, tell George. Okay. So I'll introduce dietary fiber and then we'll finish it next on Wednesday. So by definition, dietary fiber is the components of the food that we consume that we cannot digest. So plant material that we cannot digest. So you already know this. Okay, so cellulose, hemicellulose, lignin, pectin, but also resistant starch. So if we process in a way that in our food we have some resistant starch, like pasta, for example, the resistant starch goes into the dietary fiber component. So that's the general kind of definition. Now, from nutrition perspective, it goes to the small intestine and does not get complete digestion. It goes to the large intestine where the microbiome can break down that fiber. Okay, so the dietary fiber includes polysaccharides and lignin. Remember, lignin is not a polysaccharide. It is a component of the plant that is not digestible in our body. That's why it belongs into the dietary fiber. So there are soluble and insoluble fiber, and they both each have different physiological benefit. Because of their different physiological benefit, I'm going to ask Kendall, can you remind me what's the physiological benefit of soluble fiber versus insoluble? If you don't know it, that's fine. I always mix them up. Fine. Put under the spot, Kendall, sorry. Fine. What I remember, you can have it correct or not, but if this one you it out. Soluble fiber helps with bulkiness because they're soluble, so they retain water and gives you that satiety. Right? Delay the digestion process and reduce satiety. Insoluble, so if you're constipated, it helps. Right? Okay, I got it. Fine, good. So because of that, we need to not only determine total dietary fiber, we need to know how much of that total dietary fiber is really soluble fiber versus insoluble fiber. They have different physiological benefits. So oftentimes we determine soluble, insoluble, and total dietary fiber. So the soluble are mostly your pectin and the hydrocolloids and gums. So for example, beta-glucan is a soluble dietary fiber. Insoluble fiber, cell wall material, cellulose, hemicellulose partly can be insoluble and sometimes, depending on its structure, can be soluble. Oh my gosh, it's so complex. And lignin, these are the ones that are insoluble mostly. Okay. I lost you? All right, it's time to lose you. All right, I will continue. Good. I stopped where I should. I'll see you on Wednesday. Rob would at the end will have